So Anixa is a clinical stage biotechnology company focused on the treatment and prevention of cancer. Anixa's vaccine portfolio utilizes a new molecular mechanism of action to prevent the onset of cancers such as breast cancer and ovarian cancer. Anixa's therapeutic portfolio consists of a cancer immunotherapy, which uses a novel type of CAR T known as a chimeric endocrine receptor T technology, which you've probably heard a lot about in the non-Hodgkin space, and recently with melanoma with progression on immune therapy, which is huge. I mean, just blockbuster of an update. And to present this kind of exciting, more novel things that I'm interested to hear personally is Dr. Amit Kumar. Dr. Kumar is an experienced scientist, executive, and former venture capitalist. He is currently chairman and CEO of Anixa Biosciences, and he sits on the boards of other public and private companies. He served on the national board of directors for the American Cancer Society from 2016 to 2022, where he led the effort to establish the BRI Edge Fund, the venture arm of the ACS. Dr. Kumar was educated at Stanford University, where he received his educated at Stanford University and received his PhD from the California Institute of Technology, and followed that with a postdoctoral fellowship at Harvard University. So, very humble to have you. Thank you, Dr. Amit. This could be our last one before lunch.
Yeah, thank you. It's always dangerous to be the presenter holding people back before lunch. So I'll try and be relatively quick. Thank you, Chad, for having us and inviting us here, and thank you all for attending this call. And thank you for that wonderful introduction. Let me begin by telling a little story. A few years ago, I was speaking at a conference, not unlike this one, about cancer research, and I met a woman who was a cancer survivor, and it was wonderful that she was a survivor. And she told me something that completely changed the way I think about cancer. As a researcher, we're always thinking about curing a person's cancer, even though curing is not something that we clinically like to use as a term. But when people get cancer, we want them to live. And that's how I always thought about it. But what she told me was that she was incredibly happy that her cancer had been addressed. They had caught it early, and she was fine at the time. But for the rest of her life, she would wake up every morning thinking and worrying that it was going to come back. The anxiety was going to be there forever. Once you have cancer and once you've survived cancer, you're always at greater risk of a recurrence than someone who's never had cancer and getting primary cancer. So that completely changed the way I thought about it. She would get up. She told me she would get up every morning. If she had a headache, she was worried that it had come back and spread to her brain. If she had a pain in her abdomen, had it come back and spread to her liver or somewhere else like that. So again, that changed the way I thought about cancer. And then when I got involved with this company a few years ago, I said, let's try and address things like that. And so I'll tell you a little bit about some of the things that we're doing. So let me begin with, as a publicly traded company, just our standard disclaimer statement, which is, excuse me, very similar to the disclaimer statements of other companies. Just a little bit of background about our company. If you take a look at this company, you'll find that it's been around since the 1980s. However, I got involved about 6 years ago as a CEO of this company, and we repositioned it. It was a company that had been founded in the 1980s that was working in electronics and all sorts of other things. And I had sort of retired from full-time work and got involved with this company and repositioned it as a biotech company. So in reality, we're only a 6-year-old company. And at that time, the company had been delisted, was on the pink sheets, market cap was less than $10 million. And we spent a good 5 or 6 years developing these products that I'll tell you about, 2 of which are in the clinic, others in the pipeline, and addressing some of the concerns I told you about relating to that conversation I had with this patient. former cancer patient. So you'll see that the company now has a very robust pipeline, strong clinical data of the products that are in the clinic, and good partnerships, a number of characteristics which are very unique to biotech companies. We have strong financial position. Unlike many biotech companies, we're not raising cash. We don't need to raise cash. We're well financed for quite some time. And as I noted, excuse me, you'll see that I'm running around wearing a mask today because I have a little bit of a cold and I don't want to infect everyone. Hopefully my voice will hang in there. So we have a number of products that we're developing, one of which is the breast cancer vaccine, which is in clinical trials at a single site right now at the Cleveland Clinic. This is a breast cancer vaccine that's designed to prevent breast cancer. As I noted earlier, as researchers and clinicians, we're always thinking about curing cancer. But what's better than curing cancer? Not even enabling it to ever occur, right? So preventing cancer is even better than curing cancer. And that's what we're trying to do. People have tried to prevent numerous types of cancers in the past using vaccines, just like we've used vaccines to prevent infectious diseases. But that has been largely unsuccessful. And we have a theory about why that is. And we are addressing prevention through a very novel and unique molecular mechanism that has never been tried before and that we think is going to be the way people are going to eventually prevent not only breast cancer and ovarian cancer, which is one of our other products, which is preclinical, but also many other types of large incidence cancers. And we also have a product that is a therapeutic, again, addressing ovarian cancer, but this is for women who have failed all other approved therapies, and we're trying to use a unique type of immunotherapy called CAR-T, chimeric antigen receptor T-cell technology, which I'll break down in a very simple manner. I may not be as good as Sanjay at explaining very complex things, but I'll try and explain it in a manner that is relatively understandable. Thank you. Thank you, Eric. Okay, so let's talk about this breast cancer vaccine, which is our lead product, and the product that if anyone has been somewhat familiar with this company over the last year or so, we started presenting data on this product, and it's generated a lot of excitement. And I encourage any of you, if you want to go to our website and look at some of the media interviews that we've done, not only myself, but one of the key patients that has been in this trial talks about her experience. So I'm going to get a little scientific here, but at a very high level, so just stay with me for a second. So typically, when you're developing a vaccine, you want to enable the immune system to target a protein or a number of proteins that are on the cells that you want to destroy. So that's relatively easy, with a few notable exceptions, for an infectious disease, which is something that comes from outside the body into your body. Now, those infectious diseases typically have proteins that are very different from self-proteins, proteins that you see in your body. Okay? So it's relatively easy to convince your immune system to go identify smallpox or, more recently, COVID, and try and destroy those particular cells that are infected by that virus or that bacteria that's in your body. Cancer arises from your own cells. So the difference between a cancer cell and a healthy cell is somewhat limited. And so often the immune system can recognize the cancer cell to some degree, but it's much harder than trying to recognize something that came from outside your body. Now, people have tried to convince the immune system to recognize cancer cells. The way they do it is, and what they've done in the past, and there are some vaccines and clinical trials right now utilizing this mechanism that we think are going to fail eventually. And what they do is, it's well known that cancer cells express certain proteins at a higher level than healthy cells. However, those proteins exist on those healthy cells, in any case, albeit at lower levels. So what happens is when you create a vaccine targeting that particular protein, you may be able to destroy the cancer cells, but you also create autoimmunity against a lot of healthy cells. And so for the rest of that patient's life, you've got this autoimmune function that's going on that's harming healthy cells, and that could create many, many consequences later on. So, and that may be okay for a therapeutic that targets that protein, because if you start seeing side effects with that therapeutic, you can just withdraw the therapeutic. But once you've created a vaccine, once you've used a vaccine and created that immune response is always going to be there for the rest of that patient's life, potentially. So how do we address this? Well, we identify what we call retired proteins. These are proteins that exist at certain times in life to affect a certain function for the body. In the case of breast cancer, the retired protein that we've identified is alpha-lactalbumin. And the name is not important. The key is that it is what's known as a lactation protein. It enables women to lactate. So it's never produced anywhere in the body except the breast of women, and it's only produced when a woman needs to lactate. So a woman gets pregnant, gives birth to a child. The body says, I need to make this protein so we can feed the infant, okay? So the protein is expressed at that time. It enables lactation, production of mother's milk. Infant is fed. Once the woman stops breastfeeding, the protein disappears again until the woman has another child and so forth. Eventually, that protein is never seen again once a woman no longer is going to have children or reaches an age where she is not going to, is unable to physiologically have children, say at the age of 40 or 45 or something like that. However, the discovery was made, so we call that a retired protein. It's gone when that function is not necessary. However, the discovery was made that that protein comes back when cancer cells arise. So the hypothesis here, and this hypothesis was made by a brilliant immunologist at the Cleveland Clinic who's been studying this process for over 2 decades. And sadly, he passed away a few months ago, last year, at the age of 74 from cancer, ironically. But his technology, we think, is going to go well into the future. Because this protein retires from expression, meaning production, and comes back when cancer arises, we thought if we could immunize a woman after she's no longer going to have children, in principle, you're not creating autoimmunity against anything in that woman's body. All you're doing is you're teaching her immune system to destroy cancer cells as they occur. And we all know that cancer starts out as one bad cell, then becomes 2, 4, 8, so forth, and then eventually becomes a tumor mass that you can see in a mammogram. So the idea here is that we train the immune system to be ready on the lookout for a cancer cell, just like we train the immune system to look out for smallpox or COVID or something else, in an infectious disease situation. So when the first cancer cell arises, the immune system is ready and destroys it. So the woman will never know that she's getting these cancer occurrences because those 4, 8, 16 cells are destroyed and they will never become big enough to be a tumor. And so that's the vision here. And so the key here is that unlike trying to target overexpressed proteins, we're targeting a protein that should never exist in the body after a woman is no longer going to have children and only exist when cancer arises. Okay, so that's the key. So a lot of preclinical work, laboratory work was done, and a lot of this is published, but the key and compelling piece of evidence that was very exciting to me and very exciting to a number of government funding agencies that are funding a lot of this research was this graph that's shown on the right-hand side. This is an animal study. It's a mouse study. This is a unique type of mouse model that spontaneously develops cancer, breast cancer. So these mice, after you birth them, you just leave them alone, let them grow, feed them, and they'll all develop breast cancer spontaneously. And what we did, or what the Cleveland Clinic researchers did with this model, is they immunized these mice after they were born. Half of them were immunized, half of them were given a placebo control. And what they found was that 100% of the mice that were given the vaccine remained cancer-free throughout their life, whereas all of the mice that were given the control got cancer, breast cancer. So there are two key points here. Number one, I've been doing biological research for decades, and maybe some of you have as well. What we find is that in biology, you never get 100% response on anything. But this is a unique situation where we've got 100% prevention of breast cancer. And the second point is that this theory of these retired proteins was now proven in a mammalian system. Mice are different than humans, but they're close enough that we're ready now to take this into human testing. And that's what we're doing right now. Now, this is a little bit of a busy slide with some data. I'll just highlight some of the key points here. We are testing women, in this case, who are triple-negative breast cancer survivors. Triple-negative breast cancer is the most aggressive form of breast cancer, causes the most deaths, has the most recurrences. And typically, when TNBC recurs, it's often much more aggressive than the original TNBC, and it's often metastatic. So it then becomes a battle to keep that person alive. And so we're starting off with that group of patients, and we want to prevent recurrence. So these are women who've already had their surgeries, have their adjuvant therapy, and are essentially survivors. They may have some residual disease, but they're essentially cured, at least cured to the extent that our medical system can cure them today. The recurrence rate for these women is about 42%. So 42%, not quite half, but close to half of these women are going to get the cancer again within 5 years. And as I noted, these are the women that wake up every morning and are worried about their cancer coming back. Now, these women, so that's 42% for overall triple-negative breast cancer survivors, but it's even higher for women who have residual disease. So a lot of these women potentially are going to have a recurrence. What we want to do is use this vaccine to try and prevent that recurrence. We believe that we can prevent a large percentage, if not all of those recurrences. And that's yet to be demonstrated, but the initial data shows that 100% of the women that we have vaccinated in the phase 1 clinical trial are showing an immune response, meaning their immune systems are learning to target that protein, which we think will enable their immune systems to destroy those cancer cells as they arise. 100% so far. Now, these graphs, you can see they're complicated to read, but some of the lines are tall, some of them are a little shorter. So we see different types of immune responses. Some women have relatively robust immune responses and some have relatively modest immune responses, but everyone has had an immune response. And the top graph and the second graph are looking for certain T cells, which are white blood cells. And the lower graph is looking at B cells, which are creating antibodies against that particular protein. And I'm sorry, I think some people have come in. I know they told me that they were going to come in to hear this talk. We started a little bit early, so I've already started talking about the vaccine. And some of these wonderful people are triple-negative breast cancer survivors who've come into the room. And there are a couple of other key points. One, at the maximum tolerated dose, which is the dose that we will use in the second stage of the trial, which is a phase 2 trial, we didn't see any side effects. All organ issues were fine. No fevers or myalgia, anything that you typically see with other vaccines. But the only side effect we saw was a little bit of irritation at the site of injection. And the vaccination process is 3 shots, 2 weeks apart, and then we monitor the immune response over time. So, oops, did I miss a slide? Okay, maybe oh yeah, here we go. I did miss one slide. I'll skip this slide. This is just talking about the different types of cohorts that we're doing in this trial. The data I showed you was from the phase 1, which are the triple-negative breast cancer survivors. We're also looking at, in a phase 1B, a very unique type of trial that ordinarily you never do on humans. And circumstances enable that here, in particular because we as a medical community have discovered that certain women who have mutations in certain genes are at very high risk of getting breast cancer, and specifically triple-negative breast cancer. A lot of these women are choosing, once they find out that they have those mutations, are choosing to have prophylactic mastectomies. They're removing their breasts, surgically removing their breasts, even before they have cancer, because they don't want to deal with it in the future. They want to reduce their risk. I think the most famous person who did this, or one of the famous people who's done this and gone public with her journey, was Angelina Jolie a few years ago. She was found to have these mutations. She had numerous members of her family who had died from breast cancer at relatively early ages, and so she had her breast removed, even though she was perfectly healthy at the time. What we're trying to see, what we're going to do is we're going to vaccinate these women before they have their mastectomies, and then after they have their mastectomies, we're going to look at their breast tissue. Like I said, this is not something that you ordinarily do with humans, except in this circumstance where we can get healthy, purportedly healthy breast tissue. We're going to look to see if there are microtumors in that breast tissue, and see if the vaccine is enabling white blood cells, the immune system, to surveil that tumor tissue. Very exciting scientific trial. But the data on the phase 1 that I just showed you a moment ago has enabled us to now begin planning for a phase 2. And a number of people here have asked me, right now the phase 1 is being done at a single site at the Cleveland Clinic in Cleveland, Ohio, but we anticipate the phase 2 will be done nationally, and a number of people have asked about whether we'll be bringing it to Louisiana, and we anticipate that we will have a site in this region at some point. And I showed you this data, and I talked about it a little bit, so I'll skip that slide. I don't think I need to talk about the market size for this. Everyone here probably knows someone who has had breast cancer, or has had breast cancer themselves, or has a family member who's had breast cancer. It's a big market. Initially, we're focused on cancer survivors and preventing recurrence. And there are 3.6 million of them here in the United States already, and multiples of that worldwide. But eventually, we want to be able to show that this vaccine can prevent primary onset of cancer. So these would be women who would be vaccinated, just like we vaccinate people for measles and other infectious diseases. We'd vaccinate them only after they've gone past the age of childbirth. And in that case, if this vaccine in appropriate trials demonstrates ability to prevent cancer, then in principle, every woman in the world becomes a potential candidate, just like candidates for other infectious disease vaccines. So let me take one moment and pivot a little bit and talk about how we're utilizing the same technology for ovarian cancer. Again, a large market. Ovarian cancer, as we've seen from some of the other presenters here, is one of those diseases which is very hard to detect early. And often in today's world, when ovarian cancer is detected and diagnosed, it's typically at a later stage, and the outcomes are very poor for these women. Again, we'd like to not necessarily detect it early, but to prevent it altogether. And we've identified, our Cleveland Clinic research collaborators have identified a protein similar to the breast cancer case, which is a retired protein. It shows up, it exists in women on the ovaries, but over time it disappears, and at menopause, it's gone from the cells. Yeah, 5 minutes. Okay. I thought I was going to have a little more time because we started a little early, but okay, let me go. Okay. Anyway, so the point here is we're doing the same thing. I'm not going to talk a lot about this program because it's still in the preclinical stage, animal stages, and we're working with the National Cancer Institute on the development of this vaccine. And if the breast cancer vaccine continues to show good data like it has already, we anticipate that this vaccine, from a shareholder perspective, is going to be incredibly valuable too, because it's utilizing the same molecular mechanism to prevent ovarian cancer. And longer term, we believe, and I'm not going to talk about this at all, that this approach, retired protein approach, can be utilized to prevent lung cancer, colon cancer, lung cancer, and many, many other types of cancers. So let me, I'm going to go through this relatively quickly because I've only got, Chad's giving me he's going to pull me off in a minute, in a few minutes. 8 minutes, okay. This is a technology called CAR-T technology, chimeric antigen receptor T-cell technology. And I believe one of the companies here that was going to speak earlier, but I think they fell out, was going to talk about this. So I'll explain this a little bit. This is a new type of immunotherapy where, excuse my voice, I apologize. This is a new type of immunotherapy where we take the blood cells, the white blood cells, out of a patient, genetically engineer them outside the body, and then put them back into the patient because we are able to genetically engineer them and make them better cancer fighters. Now, this technology has worked really well for certain leukemias and lymphomas, liquid tumors. But unfortunately, despite many years of trying, this technology platform has never worked really in any types of solid tumors, although we're starting to see a little bit of research activity there. And the market opportunity for this is very big. Multiple companies have sported multi-billion-dollar market capitalizations for this type of technology in the leukemia space. Obviously, solid tumors are a much bigger market. A little more science here, and you don't have to look at anything complicated here, but even though the slide is a little bit complicated, the key is that we've identified a protein called follicle-stimulating hormone receptor, FSHR, that exists on ovaries in women and only on the testes in men and nowhere else in the body. And so when we engineer a patient's T cells, we utilize a hormone that targets that receptor so that we make the white blood cells much better homing missiles to go target the ovary cells. Now, as I noted, this receptor is only found on the ovaries, nowhere else. However, it was discovered a few years ago, and this on the right-hand side is the first page of the New England Journal of Medicine article that described this. These receptors are found on the vasculature, the blood vessels of cancers that occur in other organs, including ovaries. So what does this mean? This cartoon on the right sort of describes that. What that means is that, and here's what happens. When a tumor starts getting bigger and bigger, it needs to induce what's known as angiogenesis, the production production of blood vessels, because the tumor needs to bring oxygen and other nutrients in and remove waste. And so that process enables the tumor to get bigger and bigger. Now, because it turns out that those blood vessels are expressing this follicle-stimulating hormone receptor, we don't know why, but it's there. We believe that our CAR-T cells, engineered white blood cells, will not only destroy the actual tumor cells, the ovary cells, but also destroy the blood vessels and disrupt the vasculature of that tumor. So there's this dual mechanism of action that's going to potentially enable us to be the very first CAR-T technology that's going to work in a solid tumor. And we've started this clinical trial. This is going on at the Moffitt Cancer Center in Florida, in Tampa, Florida. And we've just started. We've treated 4 patients. Everything is looking good. We're seeing some indicators of efficacy, but it's not clear with this small patient group. So let me just take 2 minutes and very quickly talk about the business model that we're executing here. And what we're doing is we're establishing partnerships with some key academic centers, research centers. We work in partnership with them, enabling us to keep our cash burn low, which is very unique to biotech companies. My previous public biotech company, which was sold to another biotech company, we had a large number of employees, a very expensive laboratory, burned a lot of cash. And that's typically the model for biotech. In our case, when I got involved in this company, we decided we were going to do it in a much more efficacious way. And so if you look at our financials, we're only burning about $6 million a year, even though we've taken products from research all the way to the clinic. And that has to do with the partnerships that we've established with world-class institutions, and we'll continue doing that. And our plan is not to build out infrastructure to commercialize these products. Our plan is to develop these products to a point where we can partner with large pharma companies that will then commercialize these products, manufacture, distribute, sell, market, all of those things that are very expensive to do. And so this is just our financials. As I noted, we're burning very modest amounts of cash, clean capital structure, no warrants, nothing that creates an overhang. We're relatively undiscovered on Wall Street because, as I noted, we repositioned the company from an electronics company into a biotech company. And now that we're in the clinic, we're starting to get some attention, and we anticipate that there will be a lot of interest going forward. We have no debt either. So a number of milestones, and then lastly, just a summary slide of what we've been doing. So how did I do, Chad? Yeah. Thank you. Yes, sir. 7 weeks after they're born. Yeah. No, they were not not only in this case, but also in other experiments we've done. When we vaccinated mice, enabled them to have a litter, the pups were perfectly normal, but they were not able to breastfeed because the vaccine had induced the immune system to destroy all those cells. And that also is an additional indication that the immune system is going to be able to destroy the cancer cells. That's correct. That's correct. That's right. That's exactly right. And that's the principle. So, I'm happy I explained that well. That's correct. Yeah, that's correct. That's a really good point. And there are some women who will get breast cancer in their 20s, and they're still at childbearing age at that point. So we will have to ask those women to refrain from getting pregnant. But you bring up a good, well, that's right. So you bring up a good point. We haven't done that study in humans, but we've done that study in animals, and we find that they will give birth, but they're not going to be able to breastfeed. And so in humans, you can feed the child with formula, but that's not a study we're going to do. And in fact, in humans, and in fact, we're not interested in trying to do a clinical trial. I'm sorry? We actually we didn't do that study. And most likely, we believe they would be able to, but we're not sure. Yes, sir. Yeah. That's an excellent question, and it's something that we've tried to figure out. But with this small group of patients, it was really hard to make those correlations. And so the answer is no, we couldn't say that those were younger patients or older patients. We couldn't say that those were patients who, for example, if you had multiple children and had lactated multiple times, did you have a different response? There are a whole bunch of characteristics, but with this small cohort of patients, we really couldn't figure that out, make any real correlations. What we anticipate doing in the second phase, phase 2, is having a larger number of patients and doing that. Now, one thing I want to highlight is that we're showing immune responses in all of the patients, but we haven't proven yet that we can prevent the cancer. That's going to happen in a phase 2 where we have a control group. We'll have 300 patients who are going to get the vaccine and 300 patients who are not going to get the vaccine. And we know that the recurrence rate in those 300 patients that are not going to get the vaccine is 42, 50, 60%. And we hope that in the patients that get the vaccine, recurrence rate is 0 or some small number. 5 years, yes. But typically, most of those recurrences occur in the third year, by 3 years. Yes, ma'am. We anticipate yes, but the study is going to be done for women who are within 3 years of their complete therapy, because we want to use an enriched population. It's hard to talk like this about women, but we want to be able to have a practical endpoint for the vaccine. If we were to target women who've never had cancer, that specific clinical trial might take 10, 12 years. And so we want to get to the data relatively quickly so we can bring this vaccine to the general public. Yes, sir. No. It is not. In fact, I didn't get a chance to talk about a lot of the business model aspects of our business, but our cash burn will go up. We've been burning $5, $6, $7 million over the last few years annually. It'll go up to 8, 9, maybe 10 or something like that, but it's not going to go to 40, 50 because of the way we've run this company.